So we have here the two pentose sugars that are responsible for our DNA and our RNA. Our DNA and our RNA, deoxyribose is responsible for DNA, deoxyribonucleic acid. Ribose is the pentose sugar in RNA, ribonucleic acid. The difference here is that deoxyribose, deoxy, is lacking in oxygen on the 2' carbon compared to ribose, which has a hydroxyl group here, an OH group here at the 2' carbon. So this is the only difference between these two pentose sugars. 2' hydroxyl group on the ribose sugar, 2' hydrogen on the deoxyribose sugar. That is the only difference. This difference results in increased stability of the deoxyribose molecule and allows it to form into these double helix structures. So, now let's talk about our nitrogenous bases. We have adenine and guanine, which are our purines, and we have cytosine, thymine, and uracil, which is only found in RNA, and thymine, which is found in only DNA, and these are our pyrimidines. Adenine and guanine, our purines, have a double ring structure. They have 2 rings. And our pyrimidines have a single ring. This means adenine can only pair with thymine and uracil, making 3 rings total distance between the chains of nucleic acid, And cytosine can only pair with guanine, 3 rings of distance between the 2 backbones of DNA. Now, no, you do not need to memorize the nitrogenous base formulas. I only want to drive home the point that purines have 2 rings and pyrimidines have 1 ring. However, I do need you to remember hydrogen bonding, remembering that it is a dashed line. There are two hydrogen bonds that form, between thymine and adenine. Also between adenine and uracil as well. And 3 hydrogen bonds that can form between guanine and cytosine. So 3 hydrogen bonds can form between guanine and cytosine. 2 hydrogen bonds can form between adenine and thymine or uracil. This is the difference between adenine and thymine and adenine and uracil. Can you tell? It's a methyl group, right there. Absent in uracil, present in thymine. Otherwise, they are exactly the same. So, in terms of base pairing, only certain combinations will be working. DNA to DNA, A to T, G to C, T to A, and C to G. Even though RNA is single-stranded, RNA can base pair complementarily with DNA, and also with RNA, if a single strand of RNA loops around itself, or when it is pairing with other types of RNA during a process later that we will discuss, translation, for example. So DNA when paired with RNA, adenine to uracil, guanine to cytosine, thymine to adenine, and cytosine to guanine. Remember, the DNA is in blue and the RNA is in red. That's why we can have thymine here and adenine here, but adenine to uracil here. RNA to RNA, A to U, G to C, U to A, C to G. RNA to DNA, A to T, because DNA is our blue and RNA is our red, A to T. G to C, U to A, C to G. The takeaway point is A to either T or U, depending on whether you are working with DNA or RNA, respectively. And G always pairs with C, and vice versa. C always with G. Now, for the sake of simplicity, when you are going to be drawing nucleotides, you can draw phosphate groups as a circle, bonds as sticks, pentose sugars, no matter whether they are DNA or RNA, as a pentagon, and your base pairs as some sort of arrow some kind of arrow stick. But you must label if they are adenine or thymine or guanine or some such. By convention, typically Cambridge's convention is that purines are longer. And they have a convex edge, and pyrimidines are shorter, and they have a concave edge. So this is either T or U, depending on the circumstance. All right? That's by convention. However, don't be surprised if you see it other ways. It has been done many different ways. So, some common spelling mistakes, things that are specifically no-nos on the Cambridge exam. Adenine is a nitrogenous base. Ribose is a pentose sugar. When you add adenine and ribose together, you get adenosine. Adenosine is the combination of adenine and ribose together. They are not the same thing. Adenosine is not the same as adenine. Do not confuse the two. Thymine is a nitrogenous base. It is THYMINE. It has nothing to do with thiamine. Thiamine is vitamin B1. Pyrimidines are cytosine, thymine, and uracil. Pyramids are found in Egypt and Mexico, most notably. Uracil, once again, has nothing to do with Duracell batteries. So, now that we've cleared that up, I would like to introduce you to a very handsome molecule known as ATP, my good friend. So, ATP is adenosine triphosphate. Adenosine, adenine plus ribose. Adenine plus ribose. Triphosphate, 1, 2, 3, tri. Mono, di, tri. Phosphate groups. So there are 3 phosphate groups and adenosine, adenine plus ribose. So, ATP is effectively the most important molecule you're going to be dealing with. ATP is the storage unit we use for pretty much every energy-intensive process required for life. It is not energy. I want to reiterate this. ATP is not energy. It is energy currency. You spend and expend ATP for energy. You break the bonds in ATP in order to obtain chemical energy, but it is not energy in and of itself. Mitochondria do not produce energy. Mitochondria produce and synthesize ATP in order to break that ATP to use and to utilize that for its chemical energy. Do not substitute ATP for energy when you are writing the exam. That is unacceptable. You will not get points. All right. So the point here for ATP is that there are anhydride bonds. These are your high energy bonds. These release an enormous quantity of energy when they are broken. Somewhere between 30 and 12 kilojoules per mole, depending on what source you do, depending on whether you're talking about just a bomb calorimeter or you're talking about certain different systems. But how are we going to get this sweet, sweet energy out of ATP? Hydrolysis. Hydrolysis is how we are going to extract this energy from the ATP molecule. The ATP molecule, these anhydride bonds, these phosphorus oxygen phosphorus bonds are going to be broken by hydrolysis. We add water. We add the hydroxyl group here, and we get a hydrogen ion here, and we are left with ADP and an inorganic phosphate. ADP plus PI. So, we start with ATP. We add water. We yield inorganic phosphate and is terrible. Let's fix this. We add we get inorganic phosphate and ADP, as well as an additional hydrogen ion. So that is the resulting reaction that occurs here. We also get a significant amount of heat. All right? So here is our simplified ATP. This is our phosphodiester bond. Let's talk now about how we combine, how we add how we add, can we cut here, actually?
Yeah, that's a good idea.
So, let's show how we get a phosphodiester bond. The condensation reaction is how we get our phosphodiester linkage, which is how we put together individual nucleotides. Here we have our ribose sugar. This is our ribose, or RNA, excuse me. And we have RNA, and we have a 3' carbon here. The 3' hydroxyl will bond with the phosphate hydroxyl group here on this phosphate over here, which is on the 5' group. So we have a 3' to 5' phosphodiester linkage, a COPOC. The COPOC is a phosphodiester linkage, C to O to P to O to C. And it is from 3' to 5' carbon. This is the 3' carbon of the ribose sugar to the 5' carbon of the ribose sugar. This also works for DNA as well. This is still a condensation reaction. Now, this is not shown here, but the energy required to make this reaction happen requires that you have a trinucleotide, the 3 phosphate group version of whatever nucleotide you are working with. So if this is cytosine and this is adenine over here, then you want to have CTP and ATP. You want to have CTP to make the phosphodiester bond with whatever is above here. You break off the two phosphate groups at the end to get just one phosphate attaching to the 3' carbon of the ribose sugar above it. And right here, to get this adenine nucleotide to attach to this cytosine nucleotide, you need a adenosine triphosphate, and you need to break off the two phosphate groups here in order to get the energy from those anhydride bonds out in order to cause this condensation reaction to move forward. So, those are called activated nucleotides. The ATP, TTP for DNA, UTP for RNA, GTP and CTP versions are your activated nucleotides. Those have enough high energy bonds in order to move that reaction forward and extend the chain that is DNA or RNA. But the condensation reaction is back, and it will constantly be reminding you, hi, I'm here. Now, let's recall a few things before we continue. Numbering carbons. The most interesting group, our ether group, our 1,4 ether linkage in our deoxyribose sugar right here. This is our 1' carbon. It's prime because there are other carbon-containing compounds. The nitrogenous base also contains carbon. So we want to label the carbons in the ribose sugar as our prime carbons here. They are our primary carbons. They're the ones that we care about. So we label them with a prime, which is that little dash right there, that little apostrophe, the prime. So our 1' carbon is here, our 2' carbon is here, our 3', our 4', and our 5' is a peg sticking off over there. Now, the points of attachment for our side groups that make up the nucleic acid. This hydroxyl group, a condensation reaction will occur that will allow for the attachment of a nitrogenous base. Condensation reaction between whatever is attached, whatever will attach this nitrogenous base, either adenine, or guanine, or thymine, what have you. Condensation reaction will occur, 1' carbon to the nitrogenous base. So the nitrogenous base attaches to the 1' carbon. The 2' carbon, don't worry, nothing's going to attach there. It's going to be either a hydrogen for deoxyribose or a hydroxyl group for a ribose sugar, and that's going to be there, and it's going to be pretty stable. Your 3' hydroxyl group, this is going to be where our phosphodiester bond will attach for the phosphate group here to this hydroxyl group here by the condensation reaction we saw before. 3' to this phosphate group right here, continuing the phosphate sugar backbone of our DNA. The 4' carbon does not have attached side groups. It is a stationary point. It is required to be there, but it does not have side groups attached to it. The 5' carbon sticks off of our main ring structure. This hydroxyl group here will form with a condensation reaction with the phosphate group. It will be the attachment point for our phosphate group. So that's our 5' carbon attaches our phosphate group. So we have the 5' phosphate and our 3' hydroxyl. Remember also that DNA is double-stranded, and base pairs are complementary when you flip them upside down relative to one another. What that means is, and we've learned this before, where is it? Go find it. We'll get there. Here we go. When we flip them upside down, we have 3' running here to 5' there. We have 3' here running to 5' there. So when you flip them upside down, adenine pairs with thymine, and cytosine pairs with guanine. So, these are anti-parallel double-stranded. That's what that means. These are anti-parallel. They run in opposite directions, and they are parallel to one another. And they are complementarily base pairing when upside down to one another. Reverse to one another. So, moving forward. And here we are. That brings us to the concept of 5' to 3' directionality. There is a direction DNA is synthesized during DNA replication. 5' to 3'. DNA will always be synthesized from 5' to 3'. Always. The base pairs will be laid down, 5' to 3'. The 5' end will attach to the 3' end. Always. This is always how you will lay down new base pairs. 3' end will be open and exposed, and the 5' carbon will come in and attach to it. That is always how you will lay down. So, if you have a 5' here and a 3' here, it doesn't matter if you flip it backwards, we can read this back and forth, it's the same strand. AA, C, T, 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 G, G, T, C, G, A. They run backward to each other, but these are the same molecules flipped backward. So you just rotate this, and it's the same. Anti-parallel stranding. Base pairs only pair correctly when they are flipped upside down relative to one another. So, when you build the complementary strand, if we're still reading left to right, it must run 3' to 5'. So, 5' here, A, G, C, T, G, G, T, 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 C, A, A, 3'. then the anti-parallel must start at 3' over here and end at 5' over here. The complementary base, A, pairs with T, G with C, C with G, T with A, etc. So, that gives us the anti-parallel double stranding. 5' to 3', 5' to 3'. We have our ribose sugars, our deoxyribose sugars, excuse me. We have our pyrimidines, which have, our purines, excuse me, which have 2 rings. Our pyrimidines, which have 1 ring. And we have our phosphate groups, which are in yellow over here. Now, let's talk about Chargaff's rule. This is named after Irvin Chargaff. He did an experiment across numerous plant, animal, bacteria species. He studied the ratios of adenine to thymine and found that they were roughly 1 to 1. He also measured that guanine to cytosine ratio was 1 to 1. The same could not be said for example, thymine to guanine. It does not have to be 1 to 1. It could be 1 to 50 for all we know. All right? So, based on these findings, his contemporaries concluded that the nitrogenous bases in DNA must be found in these specific pairs, but his research did not extend to how this pairing might occur. So this is a picture of him when he was younger. This is how he aged. And we're going to skip over the discovery of DNA by Watson and Crick. That is a video that I urge you all to watch on your own time. Now we're going to talk about the structural specifications of DNA. DNA, the actual ladder that is DNA, is 2 nanometers in width. From backbone to backbone, it is 2 nanometers in width. The length of DNA is very long. I mean, we're talking billions of base pairs here across all the different chromosomes. So we're talking about meters of DNA. But in terms of average length, every 10 base pairs is 3.4 nanometers. That is 1 turn of a DNA helix. 1 turn is 10 base pairs. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. That will be 3.4 nanometers. So, in your double helix model, you'll have a major groove and a minor groove. Now, the major groove can also be found over here, and your minor groove over here as well. Our space-filling model is more accurate, but still not perfect because, well, it's a 3D representation, not the real thing. All right. Now, remembering, of course, that A to T has 2 hydrogen bonds and G to C has 3 hydrogen bonds. So if you were asked a question, for example, of the maximum number of hydrogen bonds in a DNA molecule of 100 base pairs, then the maximum number would be 3 times 100. Because 100 base pairs, the maximum, if they were all GC base pairs, that would be 3 times 100, 300. So the minimum would be 200. 2 times 100. So somewhere in between that number, your number of hydrogen bonds is somewhere between 200 and 300. Now, I'm going to introduce to you now the different types of RNA. Messenger RNA first. Messenger RNA is made by a process called transcription. That begins in the nucleus of the cell. The DNA double helix is separated to allow for an initial complementary RNA nucleotide to pair with the template strand. Then an enzyme called RNA polymerase sets to work replacing rNTPs, ribonucleotide triphosphates, or activated ribonucleotides, next to the 3' end of the forming RNA chain. The mRNA single helix now carries the code to make protein's primary structure. Now, there's a lot of mRNA editing that happens, but I will save that for Unit 3. All right? This is an image of what is going to happen. This is your RNA polymerase structure. This is the entire, this is an artist's rendition of that structure. we have DNA entering here. We have the separation of the DNA double helix. We have our coding strand and our non-coding strand here. Our coding strand and our template strand here. We have our NTPs coming in, and they are base pairing at the, they are being built 5' to 3'. And they are complementarily base pairing with this coding strand and separating then, and the RNA is being ejected here, and then the DNA is being re-annealed with its original partner strand. All right? Now, once the chain is complete, the mRNA single helix leaves the nucleus for the cytoplasm, where we'll encounter our next form of RNA, ribosomal RNA, rRNA to its friends. rRNA is built in the nucleolus, much the same way as mRNA is. It must be transcribed as well. So when it leaves, it is met with proteins that it binds to and forms one large and one small subunit of a massive, reusable factory-type subunit called a ribosome. Because of this permanence and the size, rRNA accounts for about 80% of all RNA in the cell. Ribosomes are found free in the cytoplasm or bound to rough endoplasmic reticulum. Regardless of their location, once encountered, the mRNA will be clamped between the large and small ribosomal subunits of the ribosome, and then translation may begin. So, here are my images. I have my small ribosomal subunit, which is comprised of rRNA and protein. My large ribosomal subunit, which is comprised of rRNA and protein. I call my large and small ribosomal subunits Mr. Clampy. Mr. Clampy clamps down upon the mRNA strand. The mRNA strand, right here. Then we have here the A site, the P site, and the E site. The acceptor site for tRNAs, which we will discuss in a moment. The P site for the peptide synthesizing, for the synthesis of polypeptide strands. And the E site for the exit of the empty tRNA molecules that no longer carry an amino acid. So our mRNA binding site is down here on our small ribosomal subunit. So this is the structure. Now, here is some foreshadowing of Unit 5. Different organisms have different ribosomes, and this comes into play when we deal with antibiotics, like erythromycin or clarithromycin. They can inhibit specifically the 50S subunit of ribosomes. Streptomycin and tetracycline will inhibit specifically the 30S subunit of ribosomes in prokaryotes. They have far less effect on eukaryotes like us, because prokaryotes have 70S ribosomes, which are built of 50 and 30S ribosomal subunits. No, they do not add up to 70. 50 and 30 do not add up to 70. However, this is a measure of density, not a measure of additive size. So you have 23S rRNA strand and a 5S rRNA strand, which combines into your 50S rRNA strand subunit. So, so your proteins that get added to your rRNA give you 50S in a compacted structure density. Your small ribosomal subunits have a 16S in size rRNA strand and a 21S in size protein strand, and when they combine, you get a 30S subunit. It's a density measurement. So 50 and 30 yields a 70S ribosome. These are small ribosomes compared to our 80S ribosomes in eukaryotic organisms. Eukaryotic cells have 80S ribosomes, which have a 60S large ribosomal subunit and a 40S small ribosomal subunit. So, erythromycin, which binds to this subunit, won't bind to this subunit. So it will kill when I catch my breath, it will bind to a prokaryote ribosome, but not to a eukaryote's ribosome. It will kill a prokaryote, but it will not kill a it will kill a prokaryote, but it will not kill a eukaryote. That's not to say that there aren't side effects to taking antibiotics. There are side effects to taking antibiotics. Always consult your doctor, please. Okay. So, the third kind of mRNA is now clamped. Now that the third kind of RNA. Now that mRNA has been clamped down upon by Mr. Clampy, our RNA. We've seen the grooves labeled E, P, and A. At this point, we have the transfer RNA, tRNA. This arrives carrying an amino acid. We are familiar with amino acids. We learned about them literally yesterday. Your typical tRNA is about 80-ish nucleotides long. It's not the shortest RNA strand out there, but considering some RNA strands are really, really, really long, it's not winning any prizes for length. But here's the deal. tRNA is not a single helix. tRNA loops around itself into a hairpin turn. So it loops around itself into a T-shaped cross. Now this is a terrible image. A better image will be on the next slide. But we'll have basically an amino acid attachment site for our amino acid. We will have our anticodon down here, and that is where we will have the binding to our codon, which is on our mRNA strand. So here is the better picture. Here is our amino acid binding site, which will always have this sequence ACC at the 3' end of the tRNA. These loops, don't worry about these loops. Our anticodon will be down here. It will be complementary antiparallel to the mRNA codon. A codon is merely a sequence of 3 bases. So our mRNA, 5' to 3', 3' to 5', 5 to 3. Antiparallel, complementary. C to G, G to C, G to C. So, tRNA, their 3-unit anticodon, will bind to the mRNA's 3-unit codon only if they are complementary. That complementarity entails that A matches with U, or U with A, and C with G, and vice versa, G with C. Each tRNA sequence we make is designed specifically to carry an amino acid or to carry nothing. We can carry up to 20 different amino acids. There are 20 amino acids in our library. Or nothing at all. That becomes a stop codon. So, the mRNA is clamped to a ribosome, and the complementary tRNA carrying an amino acid is able to fit into the ribosome, allowing amino acids to chain together in a very specific order. When we have amino acids in a specific order, we have the primary structure. From there, we have the beginnings of a protein, the sequence of amino acids in a single polypeptide chain. So, this is the overall what's going on. mRNA clamped between the small and large ribosomal subunit, the acceptor site of our tRNA carrying lysine over here, U to AAA. These are complementary, antiparallel, double stranding. We have our peptide site with our growing peptide chain, valine, glutamate, glutamic acid, serine, and glycine. And CAC is still bonded here, GUG, G to C, U to A, G to C. And our E site used to be on the P site, which used to be on the A site. This one used to be carrying glutamate, glutamic acid. A peptide bond will form here. More on that in Unit 3. So the differences between DNA and RNA. DNA is more chemically stable than RNA. DNA is more durable, whereas RNA is somewhat temporary. DNA is composed of deoxyribose sugar. The pentose sugar is deoxyribose, whereas in RNA it is ribose, the pentose sugar. DNA contains thymine, and RNA contains uracil. They both contain adenine, cytosine, and guanine. But DNA contains thymine, whereas RNA contains uracil. That distinction is important and worthwhile for you to recall. Locations. DNA is found in the nucleus, in mitochondria, and in chloroplasts. Whereas in RNA, it is found wherever DNA is found because it is transcribed from DNA, but it is also traveling everywhere else within the cell, into the cytosol. So it's manufactured in the nucleus, but it travels everywhere else within the cell to be translated into protein. Now, DNA has a double-stranded structure, and RNA has a single-stranded structure. Now, there are 3 flavors of RNA: messenger, ribosomal, and transfer RNA, whereas there's just the one really big flavor of DNA, double-stranded DNA. Now, that's an assignment that we did. All right, our glossary of terms. mRNA will be back in Unit 3. Transcription. Transcription is the process of making an mRNA strand from a DNA blueprint or template. Translation is the process of making mRNA information and making a protein's primary structure. The template strand is a DNA strand that is complementary to the RNA chain that is being transcribed from. And a coding strand is the DNA strand that the completed mRNA is meant to resemble. A protein is a macromolecule that is made up of amino acids. Enzymes, more on enzymes immediately following this, but for now a gross oversimplification. It is a globular protein that has a job to do. RNTPs are ribonucleotide triphosphates. ATP is an example of this. CTP, GTP, and UTP are examples of this as well. Ribosomes, a mixture of rRNA and protein that functions as the site of translation. These are rRNA and protein. They have a large and small ribosomal subunit. A ribosomal subunit is one of 2 units, large and small, that clamp over mRNA that make up the complete ribosome. Now, this is an A-level topic that is fun for me, but not an AS-level topic. So this is how we can have activators and repressors and mediators and transcription factors that all promote the transcription of RNA from DNA. So RNA polymerase is attached to all of these transcription factors, and promoter regions are binding to these transcription factors. The transcription factors are binding to these promoter regions of DNA to help place the RNA polymerase on that region that allows for the coding of the RNA transcript. So we have enhancer regions here, we have activators and receptors that prevent the forward motion of this RNA polymerase system throughout the RNA polymerase complex down the chain to produce this RNA transcript. Now, let's talk about enzymes now. Baseline knowledge. You need to write these definitions down at home if you don't have them already in your notes. The definitions for enzymes, for the enzyme for metabolic, for anabolic, for catabolic, for catalyst, for substrate, active site, complex, globular, synthesis, denature, activation energy, pH, the bond types, covalent ionic hydrogen and hydrophobic interactions. Now, let's go through these one by one. An enzyme, as I said before, a globular protein with a job to do. This is an enzyme that has a 3D conformation. It has a specific shape and it has a specific function. That shape and that function are intrinsically linked to one another. So an enzyme performs a function. It is a protein that performs a function. Metabolic is the overall chemical reactions that a cell performs. Anabolic are the building up reactions, the reactions that build The catabolic reactions are the reactions that break down within the cell. The net of all the anabolic and catabolic reactions is metabolism, metabolic reactions. A catalyst. An enzyme behaves as a catalyst. A catalyst is a chemical that participates in a chemical reaction and speeds up the rate of a chemical reaction, but is not consumed while forming products. So it participates in the chemical reaction, but is not consumed. So a catalyst, an enzyme is a protein catalyst for chemical reactions. The substrate is the molecule or molecules that an enzyme will act upon. The active site is the space, the physical part of an enzyme where that substrate will bind in order for that chemical reaction to proceed. A complex is the combination of multiple molecules that are not necessarily covalently bonded to one another. So an enzyme-substrate complex is when an enzyme and a substrate are bound together, the substrate to the enzyme's active site, that forms an enzyme-substrate complex. They are a complex, they are bound together, but they are not covalently bonded to one another, necessarily. Globular. We discussed this definition before, but they are round-shaped in general, or they have a general 3D conformation. Synthesis reactions are building reactions. They synthesize, they put things together. Denature. To denature is to permanently alter the shape of an enzyme. Activation energy. An activation energy is the energy required to begin a chemical reaction. To allow a chemical reaction to proceed to completion, you must have the required activation energy. A catalyst will lower the activation energy of a chemical reaction. An enzyme is a protein catalyst, so an enzyme will lower an activation energy of a biological chemical reaction. pH. pH is the negative log base 10 of hydrogen ions. Now that is an indication of the relative acidity or basicity of a reaction. This is on a scale of 1 to 14, with 7 being neutral. All right? Low pHs are acidic pHs. That would be anywhere from 1 to below 7. Basic pHs are anywhere from above 7 to 14. The higher your number, the more basic. The lower your number, the more acidic. Your bond types. Your covalent bonds are a shared electron pair between atoms. That is your strongest bond type in an aqueous solution. Ionic bonds are a relatively weak bond type in an aqueous solution, but they are still stronger than hydrogen bonds. Ionic bonds are the attraction between oppositely charged particles. A positive charge and a negative charge will attract to one another. Hydrogen bonding is the result of a dipole interaction between a positively charged, a net positive charge of a hydrogen, for example, in the hydroxyl group, and the net negative charge of the oxygen in that hydroxyl group, for example. But you can also have hydrogen bonds between amine groups, for example, or between an ionic compound and oxygen. So a positive ion and the negative oxygen, or a negative ion and the positive hydrogen in water. So hydrogen bonding is a weak dipole interaction for our purposes. Hydrophobic interactions are interactions between non-polar substances. Typically for enzymes and proteins, these are the reactions that are, these are the actions or the intermolecular forces that are forcing non-polar R groups inward in an aqueous solution, inward onto the inside of a protein. Now, I need to make a statement about some common mistakes Complementary is not the same as matching. They are absolutely not the same. Complementary would be when you have this matches with this, all right? That would be complementary. They do not match. Match would be if they were both squares. These match. These are complementary. So these are complementary, these are matching. All right? So this will fit inside of here. These match. Reactants are not the same as substrates. A reactant may not be a substrate. A reactant may not bind to the active site of an enzyme. A reactant may just be a participant in the reaction, but the substrate is specifically a molecule that binds to the active site of the enzyme as part of this chemical reaction that is being catalyzed. So a substrate must bind to the enzyme. A reactant is not a substrate. A substrate may be a reactant, but a reactant is not always a substrate. They are not always the same thing. Now, word roots. Mono is one in Greek. Uni is one in Latin. Poly is many in Greek. Milli is 1,000th in Greek? No, Latin. Kilo is 1,000 in Latin. Intra is inside in Latin. Endo is inside or within in Greek. Extra is outside or without in Latin, and exo is outside or without in Greek. These word roots and prefixes and suffixes will come up as we continue forward. So, enzymes are globular proteins. They catalyze chemical reactions. A standard chemical reaction, for example, would be carbonic anhydrase. That would be an enzyme. Carbon dioxide plus H2O yields carbonic acid, H2CO3. Or we could be building monomers into a macromolecule. DNA polymerase, building long chains of DNA, forming that phosphodiester bond. We could also be transferring groups, like for example, phosphotransferases. They transfer a phosphate group back and forth between molecules. Isomerases will change a molecule from one version to another. An isomerase will change alpha glucose to beta glucose or beta glucose to alpha glucose. Typically, we're talking about beta to alpha, though, just as a general rule. We also have enzymes that can digest larger molecules. Pepsin is a molecule that digests proteins in your stomach. Lipase is a enzyme that comes from your pancreas that goes into your small intestine. That digests lipids. Amylase is found in your saliva and your pancreas. Those are for the breakdown of complex carbohydrates. Now, I want to point out something. ACE, 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 ACE. ASE is a very common enzyme ending. If you see ASE at the end of a word, you're pretty much assured that it's going to be an enzyme. You're pretty much assured that it's an enzyme, which means it's made of protein, which means it's made of amino acid. Don't be fooled by questions that tell you, amylase, it's a carbohydrate, right? It's not. It's an enzyme. It's made of amino acids. All right. So, yeah. So, enzymes will catalyze chemical processes, which means they lower the activation energy by a reaction by participating without being consumed. Different enzymes do different jobs, because enzymes will only have one job. If a reaction can be catalyzed in one direction, then it's likely that that reaction can be catalyzed in the other direction, either by the same enzyme doing its job backwards, or by a different enzyme doing the opposite job. So, enzymes are produced within the cell because they are proteins Therefore, they are made by translation. We can divide them into two groups by location of where they do their job. We have intracellular and extracellular. These terms will mean intracellular, within the cell, and extracellular, outside the cell. These enzymes, the ones that will work inside the cell, will be doing enzymatic reactions inside the cell and doing metabolic reactions and such. Extracellular reactions will be taking place outside the cell, and I'll leave it to you to come up with examples on your own. This is the internet, after all. So, let's talk for a moment about activation energy. You need to start a reaction. You need to start the reaction by introducing activation energy. Activation energy is the hump that you need to get over to start rolling down that hill. So this is your activation energy in an endothermic reaction, and this is your activation energy in an exothermic reaction. You still need to input energy, no matter what type of reaction, whether you are eventually going to absorb heat or give off heat from the reaction, you still need to input energy in the first place to get that reaction started. It doesn't matter if you have one of those heat pads that you shake up or one of those cold packs that you shake up. You still need to do that activation energy of shaking up the pack. And that is going to start your chemical reaction. Your activation energy is shaking up the cold pack or shaking up the heat pack. And that is going to start your activation, that is going to input your activation energy, start your chemical reaction, and you'll have either endothermic or exothermic reaction. So, you have the energy of your reactants here. Your activation energy is applied. Your energy of your products is above the energy of your reactants. You are absorbing energy. That means the environment around your reaction is going to lose heat. Heat will enter into the reactants to form the products. You will lose temperature, heat in the form of losing temperature. Outside the reaction, cold pack. Exothermic reaction. You have your energy of your reactants here. You apply your activation energy, and your energy of your products is lower than your energy of your reactants. You have this much energy to be released. That energy will be released to the environment in the form of heat, and that will make things warmer. This is your heat pack. Endothermic reaction, shaking up your cold pack and getting your cold pack. Exothermic reaction, shaking up your hot pack to get your hot pack, your heating. When you catalyze a reaction, you are going to take your activation energy and drop it down. So your activation energy of your uncatalyzed reaction is here. Your catalyzed reaction is here. So, the energy of your reactants is the same. The energy of your products is the same. Nothing else changes. The only difference is how much energy do you need to put into the system to get your products. How much energy needs to go in before you get your products. So this is your net energy being unchanged. The net energy of your reaction is the same. Your reactants, your products minus your reactants is your net energy. That remains unchanged. Now, what the heck is this? This would indicate that there are multiple enzymes catalyzing multiple steps in one reaction. Even if you were to take glucose, for example, and burn it in a fire, that is one big step. Lighting it on fire is the activation energy, and you get carbon dioxide and water as your products. The net energy of reaction is energy of reactants, energy of products, energy released is here. So you release heat and light, and that is your release of stuff. So let's say we're burning glucose. So burning glucose requires one big step with a match, or multiple smaller steps with enzymes to get the same product. Heat and energy. Heat and energy in your chemical system. A chemical reaction. Carbon dioxide, water, and energy. So our first step, our catalyzed reaction has a lowered activation energy here. Our second step has a lowered activation energy here. Our third step has a lowered activation energy here. Our fourth step has a lowered activation energy here. And then we can proceed to products. Our highest activation energy, our third step is our highest activation energy. It didn't have to be the third step, it just was. If our second step had a higher activation energy, it would be that one. Whichever step has the highest activation energy, that will be our activation energy with our catalyst. That will be the thing that we have to get over. That will be our minimum required activation energy for our reaction to proceed catalyzed. If we have a problem with this catalyst, nothing can happen. If we have a problem with this catalyst, nothing can happen. If we have a problem with this catalyst, nothing can happen. If we have a problem with this catalyst, nothing can happen. So if we don't have enough activation energy to get past this point, we cannot proceed. If we don't have enough activation energy to get past this point, we cannot proceed. If we have enough activation energy to get past this point, we can proceed. If we have enough to get past this point, we can get past this point for sure. But if our enzyme is broken here, then we have this activation energy to contend with in the fourth step. We have a problem. All right? Now, the mode of action of enzymes. The mode of action of enzymes, the enzyme will bind the substrate to its active site, forming an enzyme-substrate complex. The chemical reaction, coaxed into taking place by the forces within the complex, cause both to undergo a change in shape. Afterwards, the product can be released by the enzyme, and it can return to its original shape, it being the enzyme. So this is an example of an enzyme causing the breakdown of its substrate. But it could just as easily be the reverse. We could just as easily have two substrates that are being combined into one product. So this is an example of an enzyme here. We have our one substrate and our one enzyme. These are complementary. This fits into here perfectly. This is our lock and key model. Right? So our active site and our substrate, one fits into the other perfectly. They bind together. That phrasing is very important. Binding. Not bonding, binding. And we have our enzyme-substrate complex. Enzyme-substrate complex. The enzyme-substrate complex, because there are new intermolecular forces at work here, the enzyme will change its shape, and the substrate will undergo stress. That stress is mechanical energy. That mechanical stress is going to cause product formation or separation into products, or the reaction to proceed in some way, the lowering of the activation energy. If this were two amino acids, it would be pushing the carboxylic acid group and the amino group into just the right position so this amine group and this hydroxyl group are right there so water can be ejected. So twisting of these substrates into the perfect position forms our new product. Now that our product is formed, it's no longer the substrate. We have different intermolecular forces. And now it will be ejected. And now the enzyme